Hi, in this video I will talk about genetic screen using fruit flies. Now, genetic screen is just like fishing, but instead of fish, here we have genes. We fish for genes. Now, genetic screen in fruit flies could be broadly divided into three categories. One is forward genetic screen, one is reverse genetic screen, and one is modifier genetic screen. Now, forward genetic screen and reverse genetic screen are the most important ones, while modifier genetic screen is a specific type of genetic screen which is important once we know something about the signaling pathway and we can figure out the interactors in the signaling pathway. So first we talk about forward genetic screen. So forward genetic screen is a way by in the forward genetic screen what we do is we know a particular phenotype or a process and we do random mutations and see in random mutant lines where the process is perturbed or not. Then we go back and check which gene is mutated. So this is the overview of a forward genetic screen. In fruit flies, what we do, we first induce random mutations, and we know what behavior or what type of biological processes to look for. So we do a random mutation by either giving X-rays, which is pretty old-fashioned method, and now people use EMS, ethyl methyl sulfonate, which is the alkylating agent causing changes in the DNA, and titrating the amount of the ethyl methyl sulfonate, you can achieve mutations almost in the all of the chromosomes. So all of the chromosomes has kind of equal probability of hitting by this particular mutagen. So in the mutant fly, let's say all the chromosomes are hit, and then we look for the phenotypes that we are expecting. And let's say we saw a biological process is perturbed in the mutant. That means in the mutant some gene is perturbed, and that's why you don't have that biological process going on. Then we go back and check which gene is mutated. So this is the outline of forward genetic screen. So for forward genetic screen, I have a detailed video. If you wish to look at it, please click on the link below. Now, there are other type of genetic screen known as reverse genetic screen, where we know which gene we want to mutate and wanted to see that if we mutate this gene, what is the effect of it on a particular biological process or particular development phase or a particular phenotype. Let me give one example of a reverse genetic screen. One of the popular way to do reverse genetic screen is to use RNA interference technology. Now, in Drosophila, using Galfur-US system, one can achieve RNAi. People can use specific tissue-specific promoter to express Galfur, and people have battery of RNAi for different genes. Now, using this combination, people can achieve RNAi against specific genes. Now let's say here we wanted to look at the dendritic morphology of a particular neuron known as neuron X, and we think that A to Z genes could be important for the dendritic arborization of this neuron X. So what we are going to do in a spatiotemporal manner, we are going to perturb the gene A to Z individually. We are going to knock down A to Z gene. So here in this particular slide, you can see this neuron, which is neuron X. Let's say we made a Galfur, which is specific to neuron X, and here is our experimental screen. So we have a battery of RNAi lines against A to Z genes, and we have a Galfur which drives into that specific neuron known as NX neuron. Now we would create multiple crosses, take their F1 progenies, and then we are going to image their brain to check the morphology, and we would score the morphology. Let's say a hypothetical result is like this. While knocking few genes, we have increased arborization. In contrast, knocking some gene give us decreased arborization, suggesting this kind of gene B could be a positive regulator. That's why when we knock it down, we have less arborization. Whereas gene A here could be a negative regulator. And that is why when we knock down the negative regulator, we get more arborization. And we can code these kind of simple features to see which knocking out which gene can affect a process, and thereby we can do reverse genetic screen. And there are many successful reverse genetic screen which has led to identification of many important biological processes. And another type of genetic screen is known as modifier genetic screen. And also modifier genetic screen has led to identification of multiple pathways that we now study in the textbooks. So modifier genetic screen is also very important, and I will be talking about modifier genetic screen in a different video. I hope this preliminary video helped you. If you like my video, give it a quick thumbs up. Please subscribe and like. Thanks.